The DNA replication refers to the process of copying a DNA molecule. This replication process consists of separating each strand of the DNA double helix. The DNA replication is a complex process because it requires the precise and synchronized participation of several specialized proteins, known as enzymes.
Now focusing on the transcription process itself, it happens in three main stages. First, intitration takes place. During this stage, RNA polymerase binds to a specific region of the DNA called a promoter. This causes the DNA strands to unwind and separate, exposing the template strand that will be used to build the RNA molecule. Next comes elongation. Here, RNA polymerase moves along the DNA strand, adding complementary RNA nucleotides one by one. Instead of the main RNA uses uracil, so adenine pairs with uracil during this process. As the enzyme progresses, the RNA strands grow longer. Finally, we have termination. In this stage, RNA polymerase reaches a termination sequence in the DNA. This signals the end of transcription, causing the enzyme to detach and release the newly formed RNA strand. In eukaryotic cells, this RNA is not ready yet. It undergoes processing steps like the addition of 5 cap, a poly tail, and the removal of introns through splicing. These modifications prepare the RNA to leave the nucleus and participate in protein synthesis.
DNA replications in eukaryotic cells takes place in the nucleus. This is where the DNA is located and protected. The process happens during the S phase of the cell cycle, when the cell prepares to divide. Replication starts at multiple origins inside the nucleus, forming replication forks where the DNA is copied. It also can occur in the mitochondria, but mainly in the nucleus.
The finality of DNA transcription is to make the genetic information stored in DNA accessible and usable by the cell. DNA itself remains safely inside the nucleus, but the instruction it contains must be carried out to the ribosomes where proteins are built. The transcription archives this by producing messenger RNA or mRNA, which is a complementary copy of the DNA sequence. This mRNA acts as a portable message that travels out of the nucleus and into the cytoplasm. Once there, ribosomes read the mRNA code and translate it into chains of amino acids, forming proteins. These proteins are essential for nearly every cellular function, from growth and repair to communication and defense. In short, transcription transforms the static genetic code into a dynamic message, ensuring that the information of life can be expressed as the proteins that sustain it. The process. DNA transcription begins when the double helix unwinds and exposes a gene. The RNA polymerase then reads one strand of DNA and builds a complementary messenger RNA, replacing the thymine with uracil. After processing, the mRNA leaves the nucleus and travels to the ribosomes, where it guides protein production. In this way, transcription turns genetic information into a useful message for life.